Hello and welcome to Lyra Nara. Today's video marks the first part in a two-part series of videos that we're gonna do on Lyme borreliosis, an epidemic with over 300,000 new infection cases per year that is truly becoming a big worry. And not only because more and more people are becoming infected by Lyme borreliosis, but also because, as we will see and explore also the reasons why, it's very difficult to detect. It comes with many co-infections and therefore it is also notoriously difficult to treat. In this first part of the series, and by the way, I highly recommend that you subscribe to our channel and hit the notifications button so you don't miss out on the second part that's gonna follow soon. In this first part, we're gonna discuss what Lyme borreliosis has to do with spirochetes, Protozoa, and Somatids. So we're gonna gain a better understanding of what really causes Lyme borreliosis, how you become infected, and then in the second part, after we've gained this understanding, you will also see why we propose the solutions that we do. So without further ado, let's learn more about Lyme borreliosis and what really causes it. Let's kick this off by discussing why we have this Lyme borreliosis epidemic, because this is a major question that has not been satisfactorily answered until now, and is also the main reason why medicine does not offer a real cure for Lyme borreliosis, relapsing fever borreliosis, and the many co-infections that come with it. There have been, however, ideas proposed of why this epidemic exists, one of them being climate change. Climate change has nothing to do with the Lyme borreliosis epidemic that we see, however, because the capability of the spirochetes to adjust their metabolism, their proliferation, and even their own DNA to any kind of climate condition is unlimited. And therefore you can find spirochetes all around the world, from Alaska to the Sahara Desert. And in different regions, there are different infection vectors with different Borrelia strains, which have changed their DNA and characteristics to better adapt and survive. Another factor that was proposed to explain the Lyme borreliosis epidemic is the deforestation of large areas in our world and the urbanization of our life. This may be true, but only if you consider Borrelia as a disease that is only spread by ticks. In the last 50 years, other animals and insects such as mosquitoes, biting flies, mites, fleas, spiders, birds, arthropods, rodents, and even dogs and cats have been playing an important role in Lyme and the co-infections epidemic that we see. Then we have the idea that this Lyme borreliosis epidemic stems from the globalization of our food chain supply and the extensive global traffic with all kinds of merchandise. Problem here is that Lyme can be transmitted by exchanging body fluids only, and food origin has almost nothing to do with Lyme. Borrelia preferentially colonize the urinary tract, and thus urine is a medium of transmitting Lyme. And there is also no Lyme immunity, and therefore every person infected with Lyme disease is an epidemic Lyme vector. And the numbers are really worrying. The CDC accepted in 2013, under the public and activist pressure, that there are about 300,000 new Lyme cases per year. Other more recent and probably more credible estimates show that new Lyme infection cases can be and probably are up to 12 times higher than this number per year. And difficult and chronic Lyme disease sick people cannot be cured because of the overuse of resistant antibiotics, and they in turn then infect other people. And cystic Borrelia forms can survive also years in freezing and hot earth regions and can become virulent when they enter a host. In regions where Lyme is endemic, this and cystic forms are found on plants, in soil dust, and in many animals. So let's find out why some of these explanations do not cover the rapid spread and do not explain the rapid spread of this borreliosis epidemic. Where is the thinking wrong and the conclusions wrong, and why have we not come to a satisfactory therapy method? And to find an answer to those questions, we need to understand what causes Lyme disease. And this is the so-called Borrelia bacteria. And the Borrelia bacteria are spirochetes. What are spirochetes then? Well, spirochetes are tiny bacteria, and their name comes from their coiled hair appearance, as you can also see in the pictures on the right. Spirochetes are ancient bacteria which have been living on this planet for billions of years, and they are very well adapted to all weather and temperature conditions. They are very resilient, and they can survive in Arctic ice, or they can be burned at 200 degrees Celsius and still survive. Spirochetes cannot make their own nutrients and scavenge thus from their hosts, so they have a parasitic behavior. And Borrelia spirochetes get their nutrients from collagen, hard tissue, aqueous humor of the eyes, meninges of the brain, the endothelial tissue, and the glial cells of the brain. The spirochetes, they attach to the tissue and damage this to the level that they can feed on it. Many organisms within the spirochetes group cause prevalent diseases. Some important pathogenic members of this phylum include the Borrelia species that causes the Lyme borreliosis, and to those belongs, for example, the Borrelia burgdorferi, which causes Lyme arthritis, the Borrelia garini, which causes neuroborreliosis, the Borrelia afzelii, which causes dermatoborreliosis, etc., etc. As you can see, there are many Borrelia species that cause Lyme borreliosis. Then we have other Borrelia species that cause what is called relapsing fever. To those belong, for example, the Borrelia miyamotoi, Borrelia recurrentis, that is transmitted by body lice, Borrelia caucasica, Crossidurae, Dutoni, Graingeri, etc., etc. And these strains behave like malaria and Babesia parasites. And then we have also Borrelia species transmitted only by mosquitoes, for example, BR91, 149, 151, 173, 177, 193, 208, and 231. And these strains have DNA that links them to the original Borrelia burgdorferi, but also to genetically modified pathogens. And Lyme borreliosis caused by these strains manifests with the most virulent symptoms, and supposedly these strains were handmade, man-made, and bio-weaponized during the Gulf War, same as with the Mycoplasma. Borrelia vincenti is commonly found in our mouths and causes what was known as trench mouth, which afflicted soldiers in World War I. It goes also systemic, infecting the brain, the heart, the tonsils, and other organs. And this bacteria can be transmitted by kissing. Trench mouth is a severe gum infection caused by a buildup of bacteria in the mouth, and it's characterized by painful bleeding gums with ulcers. The infection worsens and damages the gum tissue if left untreated. Next, let's have a closer look at the life cycle of this Borrelia bacteria. So the Borrelia bacteria behave in their metabolism patterns and proliferation, sexual as well as asexual proliferation, by the way, like microparasites, so like Protozoa. They are very similar also to the microparasite Plasmodium, which causes malaria, Babesia, and toxoplasmosis. So Borrelia spirochetes must scavenge their nutrients from the hosts. They have parasitic behavior, as we've mentioned already. They also penetrate initially the blood and the lymph system. They attach themselves to blood vessels, walls, and to those organs the blood flows through, such as lymph nodes, the heart, the spleen, and the liver. Borrelia is always found in capillaries, postcapillaries, venules, and larger veins, rather than in arteries, which have too high shear forces. They also use different types of glues, adhesives, to attach to different parts of the body. The Borrelia bacteria also begin to slowly drag or crawl along the surface of the vessels until they reach the preferred location for their proliferation. They love to nest in the so-called endothelial cells and acar joints between the endothelial cells. And when they reach their desired or targeted tissue, the spirochetes then stimulate their own engulfment by the host cells to be able to move into the cells, and they cause the cells to create protected compartments called vacuoles in which the Borrelia spirochetes can safely reproduce. And in about 48 hours after infection, the vacuoles are filled with dozens of those spirochetes. And in this space, the Borrelia are protected for a minimum of 2 to 3 weeks against antibiotics and other agents that try to destroy them. And that's why the very popular antibiotic therapy for Borrelia infection is not very efficient. The Borrelia also target macrophages, keratinocytes, neurons, the glial brain cells, and certainly also collagen in the joints and other organs. That's why you very often, when you have this Lyme borreliosis, you have extreme joint pain. The Borrelia also target endothelial junctions where they stimulate cytokine release to loosen the junction tissue, and thereby they create a doorway deeper into the body. And from here, they can penetrate the extracellular matrix, so the ECM, newly called interstitium, that we've discussed in our last video, which exists between the cells and the body. The ECM is rich in decorin, fibronectin, and GAGs, glycosaminoglycans, such as laminin, integrins, chondroitin sulfate, dermatan sulfate, keratin sulfate, heparin, and heparin sulfate. And all of those are preferred nutrients for the Borrelia spirochetes. This ECM is a difficult niche for the immune system and for the immune cells to penetrate, and gives thereby the Borrelia bacteria protected space in which to live and proliferate. And finally, therefore, the new spirochete babies are released into the extracellular space. What is the affiliation of those Borrelia spirochetes with Protozoa then? Well, in 1948, Van Thiel expressed his opinion that spirochetes have more affinities with Protozoa than with the spirochetes bacteria. He mentions 7 features indicating this relationship, partly after Sulzer in 1931, and these features are the following. First, in Leptospira spirochete, there is no distinct ectoplasmic envelope, so no bacteria-typical cell wall. Secondly, the division takes place by transverse fission. Differently in spirochetes, by stretching and thinning of the protoplasm and then by its breaking, compared with typical bacteria where a transverse wall is formed, and most species of spirochetes are also, like Protozoa, soluble in lipoid solvents. They are best soluble in 5% bile salts and hemolytic substances. And in bacteria, this is very rarely the case. The Borrelia also uses intermediate hosts before infecting the humans, like in relapsing fever borreliosis and Lyme caused by the Dutoni spirochetosis. And lice and ticks cannot infect humans until a certain length of time has elapsed after their infective feed with Borrelia. Thus, they are comparable with mosquitoes as intermediate hosts of malaria parasites or Glossina of trypanosomes and ticks of pyroplasm. Fifth, the regular occurrence of relapses to be found in a few spirochetosis, such as relapsing fever borreliosis, to a lesser degree in Wales disease, which is a severe form of leptospirosis manifested with chills and headaches, and loose, which is better known as syphilis, and this makes one think of malaria. Bacterial diseases may relapse only in few cases, for example in typhoid fever. Sixth reason is that there is a serological property. The lytic processes in the immune sera of man and animal infected with Leptospira take place without a cooperation of complement, which is an opposite to what happens usually with bacteria. In serological tests, lysins as well as agglutinins play a role in this reaction, and this occurs with active as well as inactivated serum. Seventh reason is that spirochetes have a relationship with the Spirilla, which is a rigid spiral bacterium that is gram-negative and frequently has external flagella. Examples include Campylobacter species such as Campylobacter jejuni, a foodborne pathogen that causes campylobacteriosis. And the eighth reason is that all spirochetes are missing the dipolar differentiation, so they have an absence of a morphologically differentiated nucleus, a very thin cell wall, and specifically show a transverse fission. So most features of those spirochetes show a closer relationship to Protozoa than to bacteria. So it would be a logical conclusion to classify spirochetes within the Protozoa group as follows. Subregnum Protozoa, class Spirochetes, family Spirochetes, genus Spirochetes, and subgenus Treponema, and the subgenus Borrelia. Between 1965 and 1990, Dr. Alfons Weber extensively studied and developed his theory about the microparasitic origin of cancer and of many other chronic conditions. And he postulated and experimentally demonstrated that the real cause of cancer is the parasitic infection caused by the protozoa of a similar kind to Plasmodium that causes malaria. Dr. Weber's theory was widely supported by many studies and experiments performed by renowned biologists and doctors such as Robert Koch, Tamara Lebedeva, the Max von Pettenkofer Institute, which is today part of the Medical University in Munich, Germany, Dr. Alan Cantwell, Professor Dr. Peter Duisburg, Sir Henry Britlin, Wilhelm Reich, and many others. And Dr. Weber's conclusion was that protozoa are more pathogenic than any bacteria and fungi together and are the real cause of malaria, Lyme borreliosis, cancer, and other chronic diseases. Protozoa, Plasmodia-like, are difficult to identify in regular blood lab work because they are hidden inside of red blood cells, so the erythrocytes. However, we've already in a previous video presented how a method with which you can show in a very simple blood test these resilient culprits. And I'm going to link the video in the description box below for you. The protozoa are defined as a group of unicellular eukaryotic organisms. So protozoa are single-celled organisms with animal-like behavior such as motility, predation, nutrition and excretion, as well as sexual and asexual proliferation. And these microparasites penetrate the red blood cells, or the erythrocytes, where they proliferate and multiply. And then they practically break and dissolve the capillary vessel walls using the lysosomes enzymes, and at this stage the parasites invasively migrate extravascularly into your connective tissue matrix and the organ's parenchyma, where the organ's tissue is destroyed, creating what we know today as degenerative chronic diseases. The final stage of this protozoa infection is marked by the invasion of the stem cells, which are, of course, the first defense mechanism, and their first defense mechanism, the way in which they defend us, the stem cells, they use what is called vacuolization, which is the enclosure of the parasites in small blisters. When there are too many vacuoles, the stem cells start dividing to minimize the number of vacuoles stuffed with parasites within themselves, and this is known as mitosis and leads to hyperplasia. Once the nutrients for the parasites kept enclosed in the stem cell vacuoles have dried up, the stem cells start dividing to reduce the number of parasites per cell, or there is a stem cell apoptosis, a cell death, due to the toxicity and starvation of the stem cell. And at this stage, the microparasites are of course freed again into the organism. Depending on the organs which are invaded and the general health state of the patient, first symptoms appear in form of skin rashes, skin eczemas, joints and lymphatic system inflammation and edemas, also dysfunctions up to a total shutdown of organ functionality. And this infectious process develops in 4 distinct stages. First stage is the so-called protozoa intravascular compensated infection. So the protozoa is contained within the blood vessels. Then we have the protozoa intravascular decompensated infection phase, which still takes place in your blood vessels. The third stage is the protozoa extravascular compensated infection. So the protozoa migrates from the blood vessels into the connective tissue and into the organs. And the fourth and last stage is the protozoa extravascular decompensated infection, where the protozoa migrate into the stem cells. And these 4 stages of infection with protozoa are very similar to malaria, Lyme borreliosis, and the co-infections of Lyme borreliosis, as well as toxoplasmosis, babesiosis, syphilis, leptospirosis, etc. Which are the symptoms then for this Borrelia protozoa infection? Well, first symptom is that your blood quality changes. So the blood viscosity increases, the erythrocytes clump and lose their biconcave shape, the cell superficial tension decreases due to the changes in the electrical charge, the oxygen transport is also severely hampered, and cell death may occur. Another symptom is that the blood circulation is significantly impaired. Because the infected erythrocytes lose hemoglobin, so they go into anemic state, and the glucose, plasma protein, serum, and the cholesterol, all of these substances are now missing in the host's metabolism. And therefore, the protozoa or spirochetes infected patients have usually diabetes 2, anemia, or even leukemia, high cholesterol and globulin values. They are also cyanotic, undernourished, tired, and fatigued, and toxic. Then we also see hyperplasia, which occurs to boosted stem cells division and proliferation, followed soon by hypoplasia, and leukemia is one of the consequences of a blood hypoplasia. Also, erythrocytes and lymphocytes counts are diminishing because the dead cells and the cellular debris are swimming around in the bloodstream. The capillary permeability is reduced and the general capillary circulation is impaired more and more. The gases can therefore not be purged, oxygen cannot permeate the capillaries, and blood is poorly oxygenated as a result. And also the cellular debris cannot be excreted, which means that the patients go into a toxic state. Then another symptom is that the infected blood plasma leaks out into the connective tissue, so it goes extravascular and into the organs. And Protozoa spirochetes now can infect the lymphatic vessels and the lymph nodes, as well as the endocrine glands, the cartilages, the organs. And this is what we've discussed briefly previously, the so-called extravascular compensated stage. Also, the blood vessels will become either too dilated or too constricted or obstructed. Some are completely blocked, so therefore aneurysms may appear to support the blood circulation. Other side effects of these plasmodial infections are microinfarction, necrosis, obturation, chronic heart insufficiency, heart arrhythmia, valve insufficiency, and may go even as far as a heart attack or stroke. The spirochetes protozoa invasion in the lymphatic system leads also to a swollen, painful lymph nodes, also known as lymphadenitis. And all known symptoms for Lyme borreliosis, especially in the chronic and late stages, as well as in many of Lyme co-infection cases, are being also described for protozoa infections in malaria, cancer, and almost all known chronic degenerative conditions such as MS, ALS, Parkinson's, dementia, and all so-called autoimmune conditions. We highly recommend that you watch again our previous video titled Lyme, the Great Imitator, to learn more on this subject specifically. Link again in the description box below. When it comes to the Borrelia spirochetes and the Somatids life cycle, Canadian Professor Nasons made another very fascinating discovery. He found out that in healthy cells, the somatids living in the tissue, the blood, and the intracellular cytosol of the cell are limited to 3 development stages, those being somatids, spores, and double spores. However, when the cell environment becomes toxic, meaning acidic, these tiny microorganisms pleomorphize into a 16 viral bacterial yeast-like fungus stages. And they do this to help ferment the glucose excess and the lactic acid resulting from the malfunctioning citric Krebs cycle process. Professor Gaston Nason said, quote, "A somatid is a basic living particle. It is indispensable to life. Without it, cellular division cannot take place and an embryo cannot be formed." Somatids are polymorphic, meaning they can grow from one form to another, from a virus to a bacterium to a fungus. He said, "We were able to grow it in a culture, and that's where we observed its polymorphism in 2 cycles. First, there is a microcycle, during which the reproductive hormone that permits cellular division is developed, and this healthy 3-stage cycle is stopped by inhibitors in the blood during certain illnesses, including degenerative diseases. And this microcycle in 3 stages keeps going and becomes a 16-stage cycle during illness. Little by little, I was able to observe somatids and I extracted them from the blood. I was able to isolate it and grow it in a culture, and that's how I established its polymorphism." So, spirochetes, Borrelia, and all other bacterial, viral, and fungal diseases are part of the 16-stage cycle. For the first 2.5 billion years, microbes were the only organisms inhabiting the Earth. Gradually, they populated other life forms, including plants, animals, and us humans. And it is estimated that a number of microbial cells residing in the human body, known as the human microbiome, outnumbers by several times the number of body cells within our system. Owing to their symbiotic relationship with the human organism and their vital role in maintaining the body tissues, microbes have become indispensable for our survival. The theory that certain diseases are caused by pathogenic microbes is one of the most persistent doctrines of modern medicine. And this general conception is largely attributable to the fact that microbes are present at the site of a diseased organ. And since the activity of microbes is accompanied by swelling, fever, inflammation, pus, discharge, and pain, microbes are believed to be the cause of infectious diseases. The diagram here on the right shows the classification of microbes in relation to the 3 embryonic germ layers and the areas of the brain from where the microbial activity is controlled. And microbes are endemic, so they live in harmony with all organisms of the ecological milieu in which they have developed over millions of years. White blood cells such as leukocytes and lymphocytes support the microbial work, and governed by images of biological warfare raging within the human organism, conventional medicine interprets a rise in white blood cells, the so-called killer cells, as an immune response aimed at attacking and fighting the infection. The academic construct of an immune system envisioned as a defense system against microbes becomes entirely meaningless. In fact, the term infection itself becomes obsolete. The invention also of autoimmune diseases, in which the immune system apparently attacks itself or the body's own tissues, shows how a scientific culture can become blinded by its own beliefs. Let's sum it then all up and come to some conclusions about what we've learned today. We've learned that when a disease cannot be cured and continues to develop permanently into one direction, becoming more and more an aggressive vector epidemic, there must be a problem with the therapy offered. When the therapy is not really a cure, that means that we do not correctly understand that health condition. Lyme borreliosis and its many co-infections are not diagnosed properly to this day, and we've seen why. The good news is that our group does offer reliable and very accurate remote health assessment that shows you in detail all the microbiological burdens. We've also seen that Lyme borreliosis is caused by spirochetes, and spirochetes are more affiliated to Protozoa, a type of microparasite, like the Plasmodium that causes malaria, Babesia, Treponema, a Pallidum that causes syphilis, as well as Leptospira that causes leptospirosis, etc. All spirochetes, as we've seen, also are billions of years old, therefore they are very well adapted to any kind of climate condition or other environmental factor such as temperature, pressure, chemicals, toxins, water quality, etc. They are extremely resilient. And spirochetes are pleomorphic, that means they change their shape, their metabolism, and their proliferation habits when the terrain milieu in the host body is changing. Terrain milieu is the extracellular space, so the interstitium and the connective matrix. It's characterized by acidic alkaline pH, oxidative stress, metabolism, whether anabolic or catabolic, hormonal spectrum. Major players are also melatonin, adrenaline, supplement deficiencies. So when you get depleted due to the parasitic scavenging of your own vitamins, minerals, and amino acids, but also digestive patterns when everything switches to fermentation, as we've learned, due to excessive consumption, for example, of carbohydrates and starches and grains. We've also learned that microbes have a symbiotic relationship with the human organism, and they play a vital role in maintaining the body tissues. Microbes have become indispensable for our survival. Governed by images, however, of biological warfare raging within the human organism, conventional medicine interprets a rise in white blood cells, also known as killer cells, as an immune response aimed at attacking and fighting the infection. The response to that antibiotic attack will be, however, pleomorphic changes which make the spirochetes resistant to almost any type of antimicrobial product. And sometimes, from being harmless helpers, the microbes, in our case of Lyme borreliosis, the spirochetes, and their co-infection allies, become harmful and aggressive. We will answer the question of how to deal with that successfully in part 2 of this presentation series about Lyme borreliosis, so stay tuned. If you have any questions about this subject, please do not hesitate to contact us 1-888-773-3196. Complete contact information also in the description box below. We love to talk to you and decide together what is best for you. If you've enjoyed this presentation, please also support our work by liking, sharing and subscribing. Also hit the notifications button so you don't miss out on the weekly holistic health content that appears on this channel. And with all that being said, stay healthy. Until next time.